Hi all, we're going to get into chapter 4, which is now looking at proteins in a continuance, but they're three-dimensional structures. So we focused a lot on chapter 3 on the two-dimensional structure of a protein, and now we'll be looking at how it's held together three-dimensionally. We'll be looking in detail more into the structure and properties of a peptide bond and how it relates to three-dimensional structure, general structural hierarchy of proteins, and distinguishing types of proteins based on their structure and function. So things like fibrous proteins versus globular proteins. And then lastly, and in our second video, we'll be looking at protein folding and denaturing at a even higher level. So unlike many other polymers, when we think of polymers in an organic sense, we can talk about things like sugars or even nucleic acids. Protein molecules actually adopt a very specific three-dimensional conformation. And this conformation is very important to the protein because it is what allows it to do its job, either as being a structure of a protein or by being something like an enzyme. So this structure allows the protein to fulfill its biological function. And what we call the three-dimensional structure of a particular protein is called its native fold. So again, that's just what we call the three-dimensional structure the protein should fold into, its native fold, how it should fold to allow it to biologically function. This native fold has a large number of favorable interactions within the protein. So there are lots of different interactions going on that hold this protein together in its three-dimensional structure. Now, there is an entropy cost to folding a protein into its specific native fold because, as you remember, entropy is, we have higher entropy when we go towards disorder. So when you fold something, you're ordering it, you're making it more structured, more ordered. And so we actually are it's going to cost us entropy. Some of the interactions that are going to hold together our protein in its native fold, its three-dimensional shape, are interactions we talked about actually in the beginning of chapter 1. So think back to those non-covalent type of bonds and also other interactions we spoke about. And if you don't remember, refer back again to chapter 1. There's a whole section on these types of interactions. These interactions are key to holding the protein together. So the hydrophobic effect is one interaction we spoke a lot about, which again is not a bond, but is an effect that hydrophobic molecules will tend to cluster together to escape the water molecules. And this is actually entropically favorable for this effect, this particular effect. We also talked about hydrogen bonds. Hydrogen bonds are one of the most key bonds in holding together and stabilizing structure within a protein. And these can happen, as we'll talk more about, with interactions between the NH and the CO, so the amine and the carbonyl group of a peptide bond between other amino acids. Other types of non-covalent interactions also hold together molecules. London dispersion effects. We talked about this when we discussed van der Waals effects. This is just a medium range, weak interaction that occurs between any atoms that hold them together just based on their proximity to each other. And lastly, there are electrostatic interactions, which the main type you'll be thinking about is an ionic bond. So if we have a positive group, so remember our amino acid groups, there are positive charged groups and negative charged groups. And the reason I told you to remember them and memorize them as those two groups is because these groups can have ionic bonds with each other. They can have a positive and a negative group interact to form an ionic bond. There's also another type of electrostatic interaction we'll be talking about involving the thiol groups, which is a disulfide link. So proteins can be thought of in levels of structure, and that's how we'll be dissecting them and talking about them through this course. We've already kind of focused on the primary structure of a protein, and that is its amino acid residue. So you can think of this when we look at a code of an amino acid and we look at the sequence that the amino acids are linked together for a protein, we call that its primary structure. It doesn't tell us anything about how it is three-dimensionally shaped, but it gives us information on that particular protein. So the primary structure we define as just the sequence of amino acids that make up that protein. Next, we'll be talking about secondary structure. Secondary structure is going to occur based on how that sequence of amino acid is then conformed into things called alpha helixes and beta sheets, particularly. Tertiary structure is then the folding of those secondary structures into a basically finalized, compact, three-dimensional structure of a protein. And there are other types of linkages that allow for tertiary structure to occur. How do we take one helix and link it to another helix? Well, we're going to use some of those interactions and bonds we talked about, and that's what makes tertiary structure. Lastly, there is a type of quaternary structure that can exist in proteins. Not every protein has quaternary structure. Quaternary structure is when you have a subunit, a whole folded protein, interacting with another whole folded protein. These are separate proteins with separate amino acid chains and their own folding and their own tertiary structure that then interact with each other. And again, not every complex or protein has this, but some complexes have this quaternary structure. So again, these are the 4 types of structures we'll be going over that describe proteins.